cell. We can see that it's composed of several organelles. They all come in different colors. From our previous talk, we can see that we know that most of our DNA comes from the nucleus. So that's a big purple blob over there. But my main obsession is there are these little orange organelles, which we call the mitochondria. Right? So when we hear the word mitochondria, probably the first thing that comes to your mind is the powerhouse of the cell. Right? And that's exactly what they do. So mitochondria provide most of the energy that the cell needs in order to do its day-to-day function. Little known fact about the mitochondria, which you may or may not know, is that mitochondria also have DNA inside of them, right? And so in the animal cell, there are two main sources for our genetic material. We have our nuclear genome, and we also have really, really tiny mitochondrial genomes. These genomes are very small in size, they're very compact, and they only encode for 13 protein coding genes. Extremely tiny, right? And they are in all of our mitochondria in several copies. Next. And so when I talk about the powerhouse of the cell, this is what I consider a beautiful crystal structure this is the energy factory inside of the mitochondria. So this is the factory responsible for producing all of the energy that our cell needs. So this is the electron transport chain. It's also called oxidative phosphorylation system, or just OXPHOS for short, right? And so we can see that it is composed of several complexes, protein complexes. I've got 4 here, but there's actually 5 in the electron transport chain. Each complex itself is composed of several subunits, subunit proteins. Now you can quickly tell that these subunit proteins come in 2 different colors. You've got yellow ones and you've got blue ones. Now the yellow ones, these are proteins that are encoded in the nuclear genome. So then the nucleus makes these proteins and they come into the mitochondria. And these blue ones are actually encoded in the mitochondrial genome. So I told you that the mitochondria has 13 protein coding genes. Well, all of those 13 are actually in here, right? And so we can see that there is both genomes need to, in essence, speak to each other quite closely in order to coordinate energy production. Several studies, including some of my studies, we've shown that when you disrupt these 2 genomes, when you get them to clash through incompatibilities, that you get severe consequences to all sorts of different phenotypes. Right? So, for instance, the classic example is mitochondrial disease, where the mitochondrial DNA is slightly incompatible with the nuclear genome, and that causes all sorts of problems in energy production. And so a very simplistic way of showing incompatibilities is, for instance, if this is a complex, the blue comes from the mitochondria, and the yellow comes from the nucleus, right? And you can see that the kind of blue Pac-Man and the yellow cheese wedge fit quite nicely together. For argument's sake, if there was any mutation, and the example that I use here is a mutation in the nuclear genome, just a mutation that changes the shape of the protein, you can see that the interaction is not that close-knitted with both genomes. And so this is the key of mitonuclear incompatibilities. And so if we see throughout this whole energy factory, there are several different places where we can start getting all these different incompatibilities. Right? And so the model system that I use, I work on fruit flies, as you can see from above. I've got some fruit flies here. Just pass them around. Right? And so what we do with these flies is I don't personally go out and catch them. I would love to do that. I get friends and colleagues that go to these different places and get them for me. And what we do is once we get them, we sequence their DNA. And so one safe assumption that I can make straight away is that a fly from Portamont, from Chile, the DNA would be slightly different to flies from, for instance, Japan, right? And so with the fruit fly, what we do is that we gather a whole bunch of different strains of fruit fly, bring them back to the lab, sequence them, see what their mitochondria and what their nuclear genes are. The beautiful thing about Drosophila is that we can also genetically engineer these flies, right? And so what I can do with these flies is I can say, I want the mitochondria from Hawaii, and I want the nuclear genome from Israel, for argument's sake. And I can create a completely new fly line that has the mitochondria from Hawaii and the nuclear genome from Israel. And then I can see what happens with these flies when they have mitonuclear combinations that have never seen each other in evolutionary history, right? So we expect that if I create, I call them Frankenflies, if I create these Frankenflies from places that are very different or whose DNA is very different, I can start creating incompatibilities. And so, in essence, this is what we have. We have fly lines, the nuclear DNA here. So these relate back to your flies. The nuclear DNA in these flies is all the same. And what I do is the only thing that differs between the fly strains that I passed around here is the mitochondria from all over the world, right? And so if I measure any trait, I can measure longevity, I can measure how many eggs they make, I can measure locomotion, any trait that I can think of. If I measure the trait on these flies and I see differences between these flies, I can attribute that difference only to the mitochondrial DNA because everything else, so the nuclear DNA, is the same between these flies that you are playing with. Right? So this is a really good tool to really understand the role that the mitochondrial DNA plays and the role that the nuclear DNA plays. Now, I often get questions about, have you thought about, crisscrossing mitonuclear interactions? And the answer is yes, we're doing that at the moment in the lab. But once you start crisscrossing, it gets really complicated and the experiments get really big. So at the moment, for the sake of the master students, we just have a small panel of flies where the nuclear genome is the same. Right? And so this is some data that we've gathered actually 2 weeks ago by a really good master student that we had in the lab. The trait that we measured, or that he measured, royal we here, he measured the number of eggs that a female fly laid in 24 hours, right? And down here are some of these different mitochondrial lines. And we can see that depending on what mitochondrial DNA the fly has, the fly will lay more or less eggs, right? And so flies that are able to lay a lot more eggs, we're looking into if the genomes are more co-evolved with each other and they work better. This is under the assumption that if they're co-evolved, they'll be able to produce energy more efficiently, and they'll be able to take that energy and put it into making eggs. Right? Lines that don't make, so poor Zimbabwe down here, they don't make as many eggs, and we think that there's incompatibilities going on that are affecting energy production. Right? Now, one of the things that I haven't told you, and this is where I get a little bit more evolutionary. I'm an evolutionary biologist by training, and so my main interest is seeing how things change through time, right? And so the different genomes, the mitochondria and the nuclear genomes, have different evolutionary interests. So we all know that we get half of our DNA from our mom and half of our DNA from our dad, right? And so that's what makes us all very unique and different from each other. Now, the mitochondria is only inherited through the female lineage. So my mitochondria is the same as my mom's mitochondria, which is the same as her mom's mitochondria, right? And so only moms are able to pass on their mitochondria to their offspring. And so, and I'm really sorry guys, in evolutionary terms, you're considered a dead end for mitochondria. I'm really sorry. And so the two genomes have different evolutionary interests, right? And so I'll try and make this as simple as possible. So if we think of selection, this is natural selection, survival of the fittest. If we think of it as a sieve, right? In the mitochondrial case, because mitochondria is inherited from mother to daughter, we think of it as just a female sieve. We don't care about males, right? And so all of these little X's up here are mutations that pop up in the mitochondria, right? And so if we think of selection as a sieve, where all these mutations must interact with the sieve. Mutations that are good for females go through the sieve, and this just means that they get passed on to the next generation, right? And mutations that are bad for females, they get trapped in the sieve, because females don't want crappy mutations, right? Things get a little bit more interesting. Because this is a female sieve, and the mitochondria only cares about females, there may be the case that a mutation that goes through that is good for females might be bad for males. And there's nothing males can do about that for now, right? And so there is the case that mutations go through that are good for females and bad for males. And why do we think that such mutation might be able to go through? Well, if you think about mitochondria, we think about energy, of course, and traits that are underpinned by energy production, right, tend to be quite different between the sexes. So a classic example here is metabolic rate, right? We know that males on average have a higher metabolic rate than females. So a mutation that might try to lower metabolic rate in females might actually be quite harmful for males that need a higher metabolic rate. So these mutations, we call them sexually antagonistic, right? They're the opposite in each sex. Another very interesting trait that has to do with energetic underpinnings is aging. So you don't need to worry about the dashed lines, you just worry about the colors. Across most species, we know that aging is a dimorphic trait. The sexes differ. And so for our species and for fruit flies, females live on average 20% more than males, right? And so because of these traits that are underpinned by heavy metabolic demand tend to differ between the sexes, we think that not all, but some mutations that go through this sieve into the mitochondria might be good for females and bad for males. And so if we think about this through an evolutionary timescale, I'm not talking about one generation or two generations, I'm talking about hundreds of generations, right? We predict that a lot of these mutations that are good for females and bad for males should stick around in the mitochondrial genome, right? And so we predict that males should accumulate all of these bad mutations. Don't worry, males, there's a silver lining in this story. And so this theory, I'm not the only one obsessed about harming males. This is actually a hypothesis which we term mother's curse, right? And it just means that the mother is cursing her sons with crappy mitochondria, right? And so, again, it involves the asymmetrical selection of the mitochondrial genome because it's passed through the females, and it results in the accumulation of male-harming mutations. Right? With any hypothesis, of course, we need proof. And so we've been working in the lab trying to gather proof. So this is work that was done in the lab where I did my PhD. And it was one of the first examples, or the first bit of evidence for mother's curse. So what they did here is that they used very similar flies to what you're handling. So the same nuclear background, different mitochondria, and they looked at what happens to the gene expression, what happens to the nuclear gene expression when you only change the mitochondria. And what they found was actually really interesting. They found that by just changing the mitochondrial DNA, 10% of the male nuclear genome went haywire, right? Was differentially expressed. But when we looked at the female genome, only 2 genes were differentially expressed. And if you looked, so if you got these 10% of the genes, we asked, what do these genes do? Most of these genes, so here we have a whole bunch of different tissues, most of the genes were actually related to male reproductive function, right? And so this was one of the first evidence that the mitochondria was actually affecting males in a negative way. Now, some of my own work that I've done throughout the years, I didn't look at gene expression, I looked at the classic longevity trait. And so this is, on average, how long do these flies live for, right? Again, these are all the different haplotypes which you should have floating around. So I looked at aging patterns for females, and I looked at aging patterns for males. And the first thing that we can see is that, on average, females live longer than males, which is what we predicted. Now, we don't find a lot of variation in females, so changing the mitochondrial haplotype didn't really have that big of an effect on females, except for this one down here. I could almost draw a straight line, right, through the means. Now, if you look at the males, they're actually bounce up and down a lot, so depending on which mitochondrial haplotype that particular male has, they will live a longer or a shorter life, right? So again, this is mitochondrial variation affecting male and not female traits. And so the big question is, are males doomed? And the answer is no, which is really good. Mother Nature has come up with wonderful ways of tackling mother's curse. I mean, if mother's curse is true, then males would basically go extinct because their mitochondria would be full of crappy mutations, right? And so there's several different hypotheses floating about. The one that I am researching at the moment is looking at compensatory mutations, right? So how do we resolve mother's curse? I believe that the nuclear genome is able to compensate for bad mitochondrial mutations that only affect males. And so again, if we have this sieve, lots of mutations, and you have this mutation here that is good for females and bad for males, so the mitochondria here acquires that mutation. We believe that if the mitochondria acquires a bad mitochondrial mutation, what will happen in evolutionary time is that the nuclear genome will evolve a counter mutation to compensate for that detrimental mutation, right? And this is work in progress. We've written a couple of papers proposing this theory, and now for the next 3 to 4 years, we got some funds to test these hypotheses, again, using this fly system. And so I just want to wrap this all back to this electron transport chain picture. And this is basically showing you the beautiful co-adaptation of two genomes that come from two very separate evolutionary histories. And hopefully I've shown you what happens when these two genomes clash. And so this has several big implications. Hopefully I've been able to convince you that the mitochondria is actually quite important. Not many people think that mitochondrial DNA is important, so hopefully you'll go back flying the flag for mitochondrial biology. Understanding these dynamics between these two genomes is extremely important, especially nowadays where we're coming into the era of personalized medicine, right? And so to treat all sorts of diseases, we sequence the human gene, the nuclear genome, but we should also start looking at associations with the mitochondrial genome. And so by understanding what the genotype of both nuclear and mitochondrial patients have, we might be able to better treat especially metabolic disorders. And of course, this has very important ramifications for new fertility technologies, which we'll hear all about in the next talk. I'm not sure if you're aware, but the UK has passed a bill allowing mitochondrial replacement therapy, and this is to treat people with mitochondrial disorders, where you basically remove the mitochondria from an embryo, the mitochondria that has a very bad mutation in it, and you replace that mitochondria with mitochondria from a third party, right? And so this is really good that the mitochondrial DNA is becoming more and more popular. And with any scientific endeavor, there's a lot of people that need to be thanked. I haven't done all of this work. This work has been done by myself and all these people. So, and I also want to thank you for listening to my ramblings for the past 10 minutes.
Do we have any questions?